Hello everyone, welcome. Thank you for joining us. We're here for our latest edition of our Rosalind Slow Run series with me. These are an opportunity for me to come back after, nearly 15 years to a platform I co-founded and a lot of problems that I had a big hand in creating. So it's a lot of fun. I've already seen a couple times that there are some opportunities for me to refresh some of my knowledge and abilities in certain areas, especially around math, which has been fun, as well as just to be bewildered by the fact that you think your memory is infinite, and then you come back to a place you've been at before and think, have I been here? So those are great. I love today's problems. Say hi in the chat if you are a return guest, or especially if you're new. As always, let us know where you're joining us from. It'd be great to say hi. I pinned some helpful links in the chat, especially if you're new. And the other thing that's important to know is that we always have starter code in the description to the videos. So take a look at our description, and there are a couple links there. We have completed code for every problem that we go through, and we'll be building that out, and then we have starter code as well. Today we're going to be looking at some challenges that are inspired by probability and genetics. These are going to be not the longest coding questions, but they're going to, for us to get them correct, we need to make sure that we understand exactly what's going on. So that is a great theme for today's era. In today's era, code is cheap, but problem solving still has a lot of value. And what you're going to see is a lot of our, the time that I actually spend coding compared to the time I spend explaining things and walking through thought processes, that ratio is going to be pretty small for today's slow run, and I think that's a good thing. Rodney, welcome from Kenya. Thank you for joining us. I'd really like to get to Kenya. I don't know when it's going to happen, though. I doubt Rodney is sitting there thinking, I really want to get to Pittsburgh, Pennsylvania. But maybe. You never know. You can dream. Normally how these work is I'm going to go through kind of the explanation of the entire question. And if you have any questions about what we're doing, feel free to chat those. Let me know. Or if you see me making a typo or an error, by all means, let me know as well. So help me along if you can. But I tend to answer questions about problems in between questions. So we'll do an entire question, and then if there are any questions, will you look at those before I continue to the next one? Iris, welcome from Vancouver. Another place I'd like to go. I think we're about ready to get started. Let me know how things are going. Hey, Pradeep, San Diego, yay. I used to live there. I miss the sunshine. We're going to get started. As always, like and subscribe, please. And we have tons of links in our video description, as well as on our channel. So join our mailing list and our Discord so that you can kind of stay in the loop about everything that we do. We have a partner project going on right now, which is Programming for Lovers. So we're pretty far along the way with that course. We're really getting into some advanced topics, but all of our streams for this as well as for that course are on our YouTube channel, so you can check them out on demand at any time. We're also going to be editing those down so that they'll be even sharper content. But the way you can stay in the loop is to subscribe for us for sure, but also to make sure that you're on our contact lists, because we have a lot more coming, and more coming from Carnegie Mellon that I want to tell you about. But we're not quite ready to let you know about all the good news that we're going to have just yet. And for now, make sure that you're in touch with us so I can let you know. All right. Anna, welcome from Cambridge, another place I used to live. Another place that many, many people might consider more desirable than Pittsburgh, Pennsylvania. Hank, welcome from the Netherlands. All right, let's get started. So we're going to look at a few questions here. The first one we're going to look at is IPRB. And IPRB, I've got a little bit of starter code from, it's a long doc string, but we have just some starter code that we have here that you can get in our directory. And we have our problem online. So here's our problem. And I'll walk through this explanation here shortly. All right. IPRB is a problem that's about genetics, but I came at creating this platform from the perspective of someone who loves math. From that perspective, I guess it's a little surprising that it had the popularity that it did. But I say that in this particular instance because this question is really one about probability. So it's a genetics question on the surface, and as we're going to see with other questions around this, there are opportunities for us to not take a full probability course, but to appreciate certain concepts that recur throughout the study of probability. So the example that we show here, and there's an introduction to Mendelian inheritance. So if you love things like Punnett squares from your foundational biological education, we have that explanation there, as well as what homozygous means, dominant recessive alleles, these sorts of things. So I'm somewhat assuming that these are concepts that you might be familiar with. But here's our friend, the Punnett square, where if you cross an organism that is heterozygous, meaning having one dominant allele and one recessive, with a homozygous recessive organism having two recessive alleles, then you have a 50% chance of an offspring that is heterozygous and a 50% chance of one that is homozygous recessive. Of course, the critical idea of Mendelian genetics here is that there is a genotype behind the scenes that, of course, is related in some way to the organism's DNA, but Mendel had no possible imagination of how this could be encoded because he was working in the 19th century. And so what we actually are able to observe is the organism's phenotype at this point in time on a surface level. So we're able to see the physical manifestation of the underlying traits that the organism possesses. So as a result, there is some probabilistic process that is choosing one of the two alleles. And we now know it's because you have chromosomes that are in pairs. So the simplest representation of a trait like this is that you have one allele on one chromosome and the other on another chromosome, and that explains why you have two. But at the end of the day, we wind up, in order to model this, what we are modeling is a probabilistic process. So here is why we frame this problem as thinking about probability. And when you work with probability, one of the easiest, clearest ways of visualizing concepts in probability that might be challenging is to place it on this sort of shared framework of something that we can easily understand and easily comprehend with our human minds. And it's for this reason that at one point in time, someone realized if we think about drawing colored balls out of a bag, this is not a process that we have any interest whatsoever about, but it is one that helps us build up intuition about things that might be more complicated to visualize, such as the process of choosing alleles on different chromosomes to form an organism. So in this particular case, we talk about, the notion that if you have two events, these two events in this particular case are thinking about a bag that contains 3 red balls and 2 blue balls and drawing a ball without placing it back. So in the first outcome, you have a 3 out of 5 chance of drawing a red ball and a 2 out of 5 chance of drawing a blue ball. And then that changes the proportions in each case, changes the number of balls that are remaining. And so if you draw a red ball, you have 4 balls remaining, 2 of which are red. And the likelihood of drawing 2 consecutive red balls then would be 3 out of 5 times 2 out of 4, or 3 out of 10. Whereas drawing a red ball then a blue ball would be 3 out of 5 times 2 out over 4, which is 3 out of 10. But drawing a blue ball and then a red ball actually still is 3 out of 10. But notice the probabilities here are a little bit different. We had 2 out of 5 for the first ball being blue. And then if you draw a blue ball the first time, you have 3 reds and 1 blue remaining. So we draw a red ball with probability 3/4, and that leads to the probability of this outcome, or a blue ball with probability 1/4, and we see that drawing both blue balls is equal to 1 out of 10. So the same principle is at play if we want to model the possible outcomes of having a population of organisms and assuming that we have a collection of organisms with known genotypes, and then being able to predict what's the likelihood that these organisms will fall in each of the possible buckets corresponding to their genotypes. We simplify this question a little bit by saying, in this particular case, we're going to assume that we've got K plus M plus N organisms, and those K, M, and N represent all of the 3 possible outcomes of being homozygous dominant. So this is like capital A, or capital Y in this case. N are homozygous recessive, meaning they possess 2 recessive alleles. And M of these organisms are heterozygous, meaning they have 1 dominant and 1 recessive allele. What the problem is asking us for is the probability that if we select 2 of these organisms and we cross them, what's the likelihood that it's going to produce an individual with something? And in this particular case, we'll say, well, we're looking for organisms that have the dominant allele, which means that we're looking for organisms that will display whatever the dominant phenotype is, because we're assuming that if you have 1 dominant allele, then you will display that dominant phenotype. Of course, we're going to assume here that we can, maybe we're working with plants, we can cross any pair of plants that we like. So there's no clear intuition then why it should be the case that if we take 6 organisms, not immediately maybe, 2 in each of these categories, that we would obtain a value of 0.783. And there's probably a mathematical explanation for this, although maybe if you change these values to be higher or more random appearing, then the number that you're going to get might be a little bit tricky. So we can then move to the level of code and start thinking about, in IPRB, how this is going to work. And here's where we may want to remember, something like how we think through these Punnett squares. So, we might have a sample Punnett square that says we have a heterozygous organism, and then we're looking at the offspring of 2 heterozygous organisms. So there are 2 possibilities. You could have an A or a capital A or a lowercase a, and then we also have another organism, and it could have either of these 2 possibilities. And so I actually need to give it a little bit more there. And so in this case, there we go. All right, so in this case we would have a homozygous dominant organism, whereas in this case we would have a heterozygous one. So what this is saying is, either of these outcomes is equally likely as which of these alleles is chosen from this organism. So you could have the dominant one or the recessive one. And the same is true for our other organism, where you could choose either the dominant or the recessive allele. And so if you choose both dominant, then you get dominant offspring. In this case, you have a dominant one from the second organism, but a recessive allele from the first organism. So we're going to have a heterozygous offspring. And then we'll have a homozygous recessive offspring in the final case. So you can see that in this particular case, as a result, you would have a quarter of your offspring on average, a probability 1/4, are going to be homozygous recessive, 1/4 are going to be homozygous dominant, and a half are going to be heterozygous, which means on average 75% of your organisms are going to display the dominant trait here for this particular trait. Now, this may be a review of introductory genetics, and it's one of the foundational ideas of all of biology, and it's something to be celebrated. It's also not correct, or I should say, it is only correct in certain cases, and there's a lot of evidence that when Mendel did his famous pea plant experiments, he jerry-rigged the data and the traits that he chose because he knew something about which traits might give him good results, which is not maybe the most scientifically kosher thing to do if we think about how science normally works, but in his case led to some pretty good outcomes. So there is a whole other part of the story about what to do when this isn't true, especially as we move towards, I'd say, trying to extend this to multiple different traits. But that's a story for another day. For today, we're going to celebrate Mendel, and we need to think about how this math is going to work. So we have a long docstring here where we establish you have K homozygous dominant individuals, M heterozygous individuals, and N homozygous recessive individuals. And if we think about how this is going to work, our idea is going to say, well, what is the first thing that we might do? In probability, the underlying idea of probability is that the probability of an event happening is kind of equal to the number of possible such events divided by the number of total outcomes. So if we just think about that on the highest level, that allows us to, in the same way that we often think about partitioning our code into multiple modular independent pieces, we can start thinking about the computation of the probability of an event as the ratio of these two things. And then that allows us to say, well, one of the things that we might immediately think about is what is, the number of possible total outcomes. And I think of this in the sense of I am selecting two organisms from this population. What is the number of possible outcomes that I could have? And in that sense, it tells me that this space of possible events is that there are N plus M plus K choices for the first organism. And it's just like drawing balls out of our bag. So in that case, I was selecting the first ball. I was not replacing it. And then I was selecting a second one. So as a result, I have one fewer choices for the second organism. Okay. So at the end of the day, one of the things that I might do is to simply go ahead and write down, when I'm implementing this, the number of organisms that I'm representing is equal to K plus M plus N. And then the number of total events or total possible outcomes that could happen, which is selecting two of these things I'm going to call that ordered pairs, and that's just the total multiplied by 1 less than the total. So this was the number of choices for the first organism. This is the number of choices for the second organism. And then at the end of this process, I'm going to return something divided by the number of ordered pairs. So I've got the denominator handled. And that's something, I'm going to call it the dominant numerator. Now, I then need to say, how is it that the dominant numerator is set? This is, in essence, the number of offspring that we expect to see, or the number of events that we expect to see, where we see at least one dominant allele in those offspring. And here is where, again, I'll go outside of the function and not think about writing this as code. This is where, in a certain sense, we might sketch this out on a whiteboard to say, what is it that we care about? You know, we care about the events or the outcomes corresponding to dominant alleles. And in this particular case, well, you have squares that look like this, but you also have squares that might look like something like this or something like this. And so we need to take into account all of those possible cases. So then we should enumerate or list out what are those possible cases that we would see. So there is the possibility that you have a homozygous dominant organism with another. And I think 3 of these cases, maybe the easiest cases for me to look at, are the ones where one of the organisms is homozygous dominant. So I'm going to look at all of these cases. And the reason why these cases are easy is because at the end of the day, I'm looking for offspring that have any dominant allele. And if one of your parent offspring, or if one of your parent organisms, has 2 dominant alleles, then you know that one of those is going to get handed off to the offspring. And so in each of these cases, you have 100% chance of passing that on. So if I want to count the number of possible outcomes that could happen in this case, well, imagine that, you know, this process occurs. We'd say, what's the likelihood of grabbing one homozygous dominant organism and then grabbing another? So if I do that, I have k choices. I'm not asking for the likelihood. I'm saying, what's the number of events here? It's k choices for the first organism. And after that, I have 1 fewer ball of that color. So then we have k minus 1 choices for that second organism. And I'll also say there's a 100% chance of dominant offspring if that happens. Why? Because that first organism has 2 dominant alleles. In each of these other cases, you still have a 100% chance, but we want to enumerate again how many of each of these outcomes there could be. So in this case, it's very tempting to say that there are k choices for the first parent, and then after I've chosen that parent, I say there are also, what is the parameter that we use for heterozygous? It is m. So there are m possibilities for the second parent, and also there is a 100% chance. So finally, we have k choices for the first parent, and n is our other parameter, and there's a 100% chance there too. That's very close to correct, but it is not correct. And the reason why is that there's 2 possible outcomes, 2 possible ways in which we could obtain, one parent being homozygous dominant and the other being heterozygous, and the same for here. In this case, it's not true. But in either of these two cases, I need to consider the possibility that when I pick a ball from the bag or I pick an organism from this population, the first organism that I pick could be homozygous dominant. And then the next one needs to be heterozygous. But it also could be the case that I first pull out a heterozygous organism and then I pull out a dominant organism. Those two cases constitute the same number of possibilities. There are k times n possibilities in each camp. So as a result, I'm going to say, let's multiply our count by 2 so that we're not undercounting and only counting half of the possibilities that we see. In this third case, I should say, the same thing applies. So I need to make sure that I multiply that k times n value by 2. So those cases are handling, different directions in our decision tree. But another direction that I could go is to say, A. And this is not something we want to consider, okay? Because that cannot produce an organism with the dominant allele. And that means that we really only have 2 other considerations. One of these is what happens when you have a heterozygous organism with another heterozygous organism. And the final one would be a heterozygous organism with a homozygous recessive organism. So either of these could produce an organism with the dominant allele. I gave you this particular Punnett square as a sample data set so that you would already know that the likelihood here is a 75% chance of producing an offspring with a dominant allele. So as a result, we know that then we're going to have a 75% chance at the end of this. But in the meantime, I need to say, well, how many possibilities are there? So I'm going to choose the possibilities, and then I will multiply each of those possibilities by the likelihood that when we cross them, you'll obtain an offspring with the desired dominant allele. So I have 2 heterozygous organisms. I know that there are m of them, and so I will say the number of possibilities is to pick m for the first, leave it out, and then pick another one from the m-1 remainder. So there is a 75% chance here. And then finally, we have our heterozygous organism with a homozygous recessive organism. That's actually the case that we saw on the Rosalind website earlier with our example. And this organism is always going to give you the recessive allele. This one will give you a dominant one with likelihood 50%. So we're going to choose a heterozygous organism. We're going to choose a homozygous recessive organism. And then we're going to say, well, I could have chosen those 2 things in one of 2 ways, depending on which is first. And so I need to multiply by 2. There's a 50% chance here, which is nice because it means that when I take this to code, the 50% is going to cancel out the 2. So you will no longer see this, and here's a great example of understanding the final result of code and seeing where it comes from, because in the final representation, the 2 and the 50% will cancel out. So you're not going to actually see that, and if you just looked at the output here, you'd see m times n, and that might not make a whole lot of sense. So you'd say, why is this m times n, and this is 2 times k times n? The point being is that now we have all of those cases enumerated. So it's just a matter of writing our function. So we will come down to where we were before with dominant numerator. And dominant numerator here is simply going to establish making sure that I sum each of these things. And I could establish each of the elements of the sum as their own variables, but I don't think that I really need to do that. So I'm going to have something in parentheses, and we're going to start with, in fact, I'll copy them down. Okay. I'll just copy this, just so I can see it. We're going to start with the number of events in this category. So we say k times k minus 1, and then we add 2 times k times m, and we're going to add 2 times k times n as well. And we'll notice that you have a factor of a 2, a factor of a k in both of these terms. So I can simplify this. I can go ahead and simplify it a little bit by saying, let's distribute that out as 2 times k times the sum m plus n. That handles both of these cases together. We don't need to consider the case where they're both homozygous recessive, because then there's no likelihood there of producing dominant offspring. Then we have our case of 2 heterozygous organisms, which I'm going to weight by the 75% likelihood. So I'll say 0.75 times the number of possible crosses. And then finally, I'll have my plus m times n term, because as I mentioned, the 2 factor leading it is going to cancel with the 50% likelihood. So now as a result of this process, we now have our dominant numerator. And we can return our solution to the function as that numerator divided by the number of outcomes that we had set before. Again, I'll reiterate this point that the function itself is not long. And this demonstrates that the code is short, but the solution is a little bit non-trivial and worth considering and understanding. So I can run this code, and in this particular case, it's IPRB. So I'll just say, look at the terminal, and I've made a mistake because I got stuck in Mathland. I did something that I talk to students about all the time, which is Python's not quite human language. And so I need to make sure I have multiplications instead of just assuming that it understands what I'm meaning. Okay, so we see that 0.78333 is indeed printed. So we have a good sense that, maybe we can apply this to a real data set. So we're going to comment that out. And now we're ready to run. So we'll save. And I'll get a data set from the platform. And once I've got my data set, let's move it over. We're always going to call it data set. And now I can rerun my code. And here is a good sign that I need to make sure that I have the updated version of custom IO. So if you don't have that, you would get a similar result to me. But let me just make sure I have that quickly. Okay. All right, let's try that again. I've got updated custom io.py. So now I actually have the right code to parse in the data set that we're calling. It gives us an output, 0.81153 for this particular data set. And as I said, here are values of k, m, and n. That would be challenging, perhaps, to compute by hand. And yet we have a formula here that will just produce it automatically. So the notion here that we understand probability and how it works, and that is helpful for us to write code to simulate things where maybe computing everything exactly, well, we are computing something exactly, but doing so by hand or without the use of a computer might become quite annoying. So in this case, we're going to submit our answer. Rosalind's having a slow morning, but we've solved the problem. Any questions, let me know. Happy to take a look at those. Kieran, welcome from Boston. Good to have you here. I've noticed I need a new chair. I don't know if you can hear how squeaky mine has become. It was new during the pandemic. Realized that was a long time ago, huh? Julia, welcome. I don't mind if you're late. I'll forgive you eventually. But probably the best pathway to do that is make sure that you join all of our mailing lists and Discords and all that good stuff. Afia, same thing. If you have any issues with getting anything to run, Programming for Lovers is our partner project where if you ever have any struggles with running Python, many people across the course of the last 15 years have given up on learning to program because everybody told them that they should learn Python. And then they tried to install Python, and then they were like, well, it doesn't run. And that's because Python's a horrible language, which is something that you'll hear me say a lot. But probably its biggest annoying trait is that there's a million ways to install it. And then if you try to install it 2 different ways or you don't do it in the right way, then it becomes impossible to actually use. No one likes me saying that, but it's because it's true. And so we have installation about how this works on our Programming for Lovers website. So that to me is, in terms of trying to teach someone how to get started in Python, that's the easiest installation I have found. It's not like a developer-y installation, but it will help you get started. And then always come to our Discord. If you have any issues, you're going to encounter a community of learners who are here to help. All right. So, next problem we're going to look at is called IEV. IEV. All right. With IEV, we're going to talk about expectation. And the idea that a lot of times in probability, we want to know what's the likelihood of a certain outcome happening. Whereas with expectation, what we want to look at is the expected value of a variable that could be random. So what is the on average value that this random variable could take? So our example here on the platform says we roll a die, and what would our expected value be for the value on that die? So in other words, what's the average case here? And there's a formula there that you might imagine the answer should be 3.5 because that's in the middle of all the integers between 1 and 6. You might think, well, I just add them all up and divide them. And that is certainly true. So we could sum the values 1 to 6 and then divide them. But then I could change things here, and the computation might not be as obvious if I made the die weighted. So in that particular case, you would see that the die has different probabilities. And so our formula here would generalize that idea to say, take the value on the die, multiply it by the likelihood that the die comes up with that number on it, and sum those products over all possibilities. This is the start of the notion of expected value, which says take the value for each of your cases and multiply it by the likelihood of seeing that. And at the end of the day, whatever that weighted sum winds up being gives you the expected value. This allows us to generalize this concept in a lot of different directions to a lot of distributions that are a lot more complicated than rolling one die. But that formula essentially is going to hold and be extendable in each of those circumstances. This problem, though, gets at a more fundamental concept, just in probability in general, which is that a lot of times I could produce a random variable that's very complicated and say, well, what's the likelihood, for example, that if I observe a bunch of these things happening, like in sequence, a bunch of these possible events, what's the likelihood that exactly 4 of them are going to occur out of maybe 150 possibilities? What's the likelihood that 4 of them are going to occur? Or what's the likelihood that between 17 and 43 of them are going to occur? And in a lot of contexts, computing that exactly might be a difficult thing to do. And yet sometimes computing the expected number of outcomes that you would see is actually much easier. It depends on the context, but in some particular cases, that's much easier. The example here talks about, well, what if you were to roll 2 dice? And one of the ways in which you could do that would be to establish all the probabilities of each outcome. So you have 36 total outcomes. And I guess, well, you have fewer than that, because you could say, if I roll 2 dice and say, what are the likelihoods of it summing to different values? It turns out that you can look at all the possibilities and say, there's a 6 out of 36 chance that it winds up being a 7. There's a 5 out of 36 chance that the sum of the dice is equal to 6 or 8, and so forth. But if you're looking just for the expected value, meaning the average case outcome, it's equal to 7. And you can get that through our formula that we have before. But you can also get it through a much simpler mechanism. And the reason why is a concept that most people are not going to hear about unless they study mathematics at, say, an undergraduate level. And that concept is called the linearity of expectation. And it's a very powerful concept. What it means is that if I ever say, what's the expected value of the sum of 2 things that involve randomness? That one way of doing that would be to go back and think about, I've got to think about all the possibilities for X and all the possibilities for Y, and I've got to figure out what their likelihoods are, and then what are the values of X and Y in each of these cases, and then use a formula like this to compute it. But that's not at all what you would actually do. When you apply the linearity of expectation, what it tells us is that the expected value of any sum of random variables winds up just being the sum of E of X and E of Y. So you can take that E and apply it to each of the random variables that you see. In this case, it makes it obvious what the expected number should be of the sum, that the average case of what you see as the sum on 2 dice is equal to 7, because this thing is just E of X plus E of Y. But we already figured out that the expected number on average of a single die roll is going to be 3.5. So you wind up with just 3.5 plus 3.5, which is 7. And that makes computing this very easy. So the question is, how is it that that concept is going to be relevant to this particular question? What does the question ask? It says, we're going to give you 6 non-negative integers, and those non-negative integers correspond to the possible, or the number of crossings in a population possessing each genotype for a given trait. And so here are our 6 outcomes that we saw previously with IPRB, where we were computing likelihood of a dominant outcome. And that's what we'll be given with our data set. So in this particular case, we only have 3 pairs of organisms that are going to produce offspring in each of these categories. So we get one that are homozygous dominant, we get another one that are both heterozygous, and then we get one that are both homozygous recessive. So this would allow you to when you look at the return, we could compute this by hand to say, what's the expected number of offspring that will display the dominant phenotype in the next generation? So we're looking for the expected number of offspring that fall into the category of either homozygous dominant or heterozygous in the next generation. We're also going to assume that every couple has exactly 2 offspring. So if we were going to compute probabilities in a lot of different cases, then that's a separate question. But it turns out what this question is getting at is that computing the expected number of offspring displaying the dominant phenotype in the next generation is actually going to be easy. So we know that in this case, there are going to be 6 offspring in the next generation. We have 3 sort of crossings that we're doing now. Each one is going to produce 2 offspring. And the sample output in this case is going to say, well, it actually happens to match this 3.5, but for very different reasons, that the expected number here is equal to 3.5 that are going to display the dominant offspring. And you maybe can start to see how this is going to work, because what we are going to do is apply this same idea and to say the expected number is equal to the expected number in this case plus 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 this case. But remember, I said when you take the expectation of the sum of 12 cases, in this case it's 6 cases, well, I can just figure out what is the expected number of offspring in each of these cases, and then at the end of the day, I'm just going to add up all of those things. So this 3.5 comes from saying, consider each of these cases in isolation. The ones with 0 are going to give you a 0. And so in this case, that is a homozygous recessive pair. The expected number of offspring displaying that phenotype in the next generation for these is going to be 0, because the likelihood of that event occurring is 0. In this case, it's going to be 2, because the likelihood of having a dominant phenotype is 100%. So you're going to have 2 offspring. You're going to expect 2. And then I guess this means that in this particular case that the expected value is 1.5. That makes sense because we have a 75% chance of it happening and a 25% chance of it not happening. So if we look at our formula, your probability is 25% times 0 plus 75% times 2. And so it's just 75% times 2, which is 1.5. So that's how we get a 2 from here, a 1.5 from here, from this value, and then a 0 from here. What we want to do is just take that idea and extend it to appreciate this concept of saying that the expected number of dominant offspring is the sum over all of the couple types that we have of the number that we see, so the number of pairs that come from this bucket. They all are going to produce 2 offspring, and then there are 2 possibilities. It's either dominant for that pairing or it's not. So if it's not, we obtain 0. If it is dominant, then we obtain 2 offspring that have it. And so we're going to say we're going to produce an expected number of offspring for that particular category of the number of parent pairs times 2 offspring times the likelihood that we see it. So once we have that appreciated, again, once we know how this works, our function is going to be quite straightforward. I'll in fact say, let's first establish a probability of a dominant phenotype offspring for each of the 6 genotype pairings. And I'll provide that outside of the function even, because otherwise I'm going to be declaring a variable inside of this function that's pretty important. And I'm just going to establish a little list of floats, and I could also have this as a tuple, but that doesn't really matter. It's going to serve a pretty simple purpose here by saying our categories are, if we look at the website, because we don't have it in the doc string, we're going to have AAAA, so that gives us a 100% likelihood of an offspring that has the dominant trait. In this particular case, it's 100%. In this case, it's 100% because one of the parents is homozygous dominant. This is the case where you have a 75% chance that we've already seen. You have a 50% chance here, and you have a 0% chance if you have 2 homozygous recessive organisms. So as a result, we have 1.0s for the first 3 values. Then we have 0.75 for when we're performing this cross of heterozygous organisms. Then we have 0.5 for the 5th case, and then 0.0 for the final one. That means I don't have to consider doing 6 computations individually. I'm just going to do them all at once. So when I compute the total, here is where I apply the linearity of expectation, which tells us that the expected value of a sum is equal to the expected value of x plus the expected value of y. And this applies to sums of multiple random variables too. I just am not writing it out for the case that we have, which is what's the expected number of offspring that have this dominant trait that you are going to see where the sum is over 6 different possibilities. So in this case, then, I'm going to say our total is going to be a float that starts off equal to 0. This is going to be our expected value. And it could be fractional. At the end of this is what I'm going to return. And then here I'm going to say, let's range over the couples and establish the expected value using the probabilities that we set. So here I'll say for I in range over the length of the couples, which is just going to be 6. But this gives us something that we could, in theory, generalize later. And I'm going to add something into the total. So the total is just computing the expected value of each of these things in turn. And so the total needs to be the number of couples multiplied by the number 2, because this is going to tell us the offspring that we care about. And then we need to multiply that by the probability. So before we saw the expected value here is the sum of your possibilities of how many things times the probability of that occurring. And if you wind up with 0, again, then you're not going to actually factor that in. So there is a hidden term here, which is couples of I times 0 dominant offspring times 1 minus the dominant probability of I. But you multiply anything by 0 and that's not really going to be important. So I'll put this in a comment and just say there is a hidden term that we are adding this in, but it's 0. So in this case then, what I'm really interested in is the number of couples times the number of offspring times the probability that the offspring are going to be dominant. And in that case, it's just the dominant probability of I. If I can get my bracketing right. And that's it. So another short function that when I run it, hopefully we see that we get 3.5, which is the correct answer on our test. So we will go to Rosalind. We'll get ourselves a data set, and we'll put that into our folder and make sure that it's named dataset.txt. Run our code again and make sure that I actually have the parsing part for our data set uncommented. Let's try it again. All right. Now, this seems like a number that is sort of like, where did it come from? And in fact, as I said, it's fractional. So it might seem that it's like a complicated idea because we're getting an expected number that's not even an integer for a collection of organisms that are integers. But the linearity of expectation gives us the fact that computing this expected number is easy whenever you're just summing it over a bunch of categories. And that's something that comes up over and over. So I was teaching undergrads the other day, and I'm fortunate our undergrads get a lot of math. And there's a concept that we were looking at in mass spectrometry, mass spectrometry and saying how statistically valid it is to report. How do we validate reporting certain outcomes of the experiment as saying we found a match in this sample to a database? And one of the challenges is the whole thing is dependent upon appreciating that we can get at this question by saying, well, what's the expected number of hits of this quality that you would expect to see in a database that's just make-believe? So the idea there is that I say I found something in the database, but you validate that by saying if I came up with a random database, on average, how many hits would you see? And computing that thing is not that hard if you know that expectation is linear, meaning at the end of the day, we wind up writing that as the expected number of hits winds up being the expected number of a bunch of possibilities happening where they all break down as a sum. And then I got to say to the students, is there anything here we get to use? And one of them's like linearity of expectation. And I'm thinking, you're very fortunate that you've already seen that, and I am too. I'm very fortunate to say that you've already been able to see this idea, because then it's just a matter of saying this concept that frankly a lot of times, and how it was taught to me as a math student, is purely in the context of math, winds up popping up all over the place. And that's what we need in order to solve that particular question. So that's a good plug, I guess, for great ideas in computational biology, which we're going to roll out soon, because that's a story we'll tell then. So you can join us for that. All right. Let me know if you have any questions. Otherwise, we're going to keep going. Our next question is going to be LIA. Just a little more probability. A couple more probability problems, and then we're going to come back to a classic string problem that actually is very related to probability, which is why we're going to cover it today. But I wanted to make sure that we got to 5 questions today. So we'll see that here shortly. For LIA, which is going to look at independent alleles, there's a biological idea here and a probabilistic idea that are inherently connected. And the biological side of this is looking at what's called Mendel's second law, which is also commonly known as the law of independent assortment. In a previous problem, I mentioned that a lot of Mendel's work is, to say the least, dubious, because it only applies to certain specific types of traits that he may or may not have cherry-picked when he performed his experiments on pea plants, because he knew how the outcomes would turn out, and that there were other cases where certain traits would not produce such beautiful data. That relates to this particular law, which is not true for many traits or many pairs of traits. And what the law says is that each trait can be viewed independently of each other. That means that at the end of the day, when I look at 2 traits, say A and B, we have the process of choosing offspring that have one of the alleles from trait A. The same thing applies to trait B. And when we look at the results of this process, we don't see any correlation. So it's not the case, for example, that if an organism is, has the dominant allele for A as well as B, but it's heterozygous, it's not the case that its offspring at a disproportionate rate are going to have both the dominant allele for trait A and trait B. So this is where Mendel's laws start to break down, and where it took people at the turn of the 20th century working on Drosophila to establish a much clearer framework for this and figure out what the actual probabilities need to be. But for many pairs of traits, this is a good model. And so in this particular case, for example, we can look at our figure and say if we have, two organisms and we have traits S and B, then when we look at, the possible ways in which they could cross, we would get we're always going to have a heterozygous organism for trait S and a heterozygous organism for trait B. And then we produce all offspring of those, and we wind up with 16 possibilities. And what the law of independent assortment says, which again is not true for all pairs of traits, is that of the 16 possibilities that you would obtain, they're all equally likely. So it's not the case that it's more likely to have an offspring falling into this category than in some other category, right? So again, that's a story for another day, but we're going to make that assumption. And the mathematical or the probabilistic side of this is to state that the events involving the two traits, the inheritance of these two traits, are going to be independent. Independence means that we can separate these two events. So if we're looking for what's the likelihood that both of these things happen, it winds up being the likelihood of event 1 happening multiplied by the likelihood of event 2 happening. So on a very high level, what we're saying is that the events do not influence each other. They're not in any sense correlated. And so it's not the case that, so imagine, for example, if the likelihood of A happening is 0.5 and the likelihood of B happening is 0.1, we're going to say, what's the likelihood of them both happening? Well, it would be 0.05 or 5%. If these events are more correlated, well, the likelihood of them both happening, that might not be the case. Maybe A always happens when B occurs, in which case the likelihood would be 10% for that example. But if the events are independent, and that's what we are assuming that is going on behind the scenes in Mendel's second law, it means anytime I have the likelihood of two things happening together, I can simply multiply it out across each of those independent events. This is not the same thing as events being disjoint or mutually exclusive. So in that particular case, I want you to imagine that I roll one die and I have two different events. One of those events is X is even, meaning what's the value on the die, and what's the other event would be what's the likelihood that the value on the die is odd. Those events are disjoint or mutually exclusive, which means the probability of either of them happening is just the sum. So you can split those two things completely. Those events are not what we would say independent of each other. So a good example of independence would be the example that's given here. If you have X is one die roll and Y is another die roll, then you might say, what's the likelihood that the sum of the two dice is odd? So the sum of that likelihood in this case is equal to, well, the sum of two possibilities. And here's where I get to produce these two possibilities that are disjoint. So two events that can never occur together are that the value on the first die is even and the second die is odd. So that's event 1. Event 2 is that what is on the first die is odd and what is on the second die is even. So those events are not independent, they're disjoint, meaning there's no possibility where both of those things can happen at the same time. So this is nice because we have found two disjoint events constituting what it means for the sum of the two dice to be odd. If the sum of the dice is odd, one of these two things has to happen. So we can write it as this sum. But then we look at, for example, what about the probability that x is even and y is odd? Those two events are independent, okay? And that makes sense intuitively, and it's why we think about grabbing balls out of bags or rolling dice, because if I roll two dice, the word independence is doing a lot of work here. Even if we're not thinking about this in terms of a formula or placing it on a very precise mathematical framework, I could roll each of these dice. They're not going to affect each other, and their outcomes are going to be independent. So the likelihood that x is even and y is odd should therefore be the product of these two probabilities. And I can break this down as a product too. So that makes it a lot easier to compute each of these things, because the likelihood that x is even is 1/2, and this is 1/2, and this is 1/2, and this is 1/2. So each of these terms just winds up being 1/2 squared, and when we sum them, we get 1/2. So notice that we computed something, which is the likelihood that you roll two dice and that the sum is odd. And I think maybe if you roll a lot of dice, you find yourself in a casino a lot, you know this, okay? But if you don't, this is not something that you might immediately know is to be true, that it's equal to 1/2. And here we use these two separate facts, splitting something up into disjoint events and summing them, and then taking independence and using an and, breaking that down as a product, and we get a final computation. The idea is, how do we apply this to a biological context in this particular case? Where is this going to be hiding? And we're going to need a little bit more insight, and it will allow me to introduce another distribution, but this idea is going to be inherent to what we're doing. So what it is that we're asking for here is that I'm given parameters k and n, and we're going to start with one organism. I'm not 100% sure why this organism is called Tom. There's no reason to think of Tom as anything other than, Tom could be a plant. And we're going to assume that Tom, our pea plant, has genotype consisting of two heterozygous traits. And then we're going to assume that Tom produces two offspring, and there's going to be exponential growth here among this family tree, so to speak. So two offspring produce two more offspring, so we'll have 4 and 8 and 16 and so on. And each organism is always going to cross with an organism that also, or an organism that is heterozygous for both traits. So note that the offspring as they go might not be heterozygous for both traits. So it's a little bit of a trickily worded problem in that way. So it's not always the case that everybody is going to be heterozygous, but what they are going to do is produce offspring with something that's heterozygous. And then we're going to say after k generations, there's going to be 2 to the k total organisms in our population. And what's the likelihood that at least n of them are going to be heterozygous for each of these traits? So we're going to assume that Mendel's second law holds, and all that means is, I'm going to assume that some of these events are independent of each other, and that's going to allow me to split an and as a product of things, something that genetically can be dangerous for a lot of traits, but that is going to be something we're going to model here. So here's where, again, we come back to wanting to code this, but I'm not really ready to code it because I need to make sure I understand precisely mathematically, how is it that I would approach a question like this? So it's an opportunity for me to see where independence, genetics, and a little bit of mathematical insight all are going to intersect in order to give me the solution that I care about. So I'll write down some things that I know. What I want to compute is I've got a k, which is my number of generations, and then I have an n, which is what we want to ask about, are there at least n organisms that are heterozygous for both traits? So at the end, we're producing a float, which is the probability that that outcome is achieved. So note, 2 to the k could be pretty big, and n is allowed to vary. So we might wind up with 128 organisms, and we're saying what's the likelihood that at least 71 of them are heterozygous for both traits. That doesn't seem like it should be a short function or an easy computation. And again, here's our opportunity to understand how the math would drive a computation like that to produce something that's really short, and why this notion of independence actually matters. So I'll write down, the number of organisms in generation k is equal to 2 to the k. And I can't imagine that's not going to be useful. So I'm going to have my math down here. Math is fun. Okay. And that's probably going to be pretty important. So as a result, I'm going to say, let's set the population that I care about equal to 2 raised to the k. Now, once I know that, I'm going to say, let's write out, I want to know the number. What I care about is the number of organisms in the kth generation that are, I could ask this in any way I like, but it's asking me for heterozygous for both traits. So x is going to be equal to this random variable, which is the number of heterozygous organisms for both traits in the kth generation. Key point, we assume that the traits are inherited, meaning A and B. They're inherited independently by this law of independent assortment, which is why the problem is called LIA. There is another little tricky point, though, which is the key to solving this problem, and why I've been impressed that thousands of people have gotten it right, which is that if one parent is heterozygous for a trait, then it doesn't matter what the other parent genotype is. Okay. What do I mean by that? So I want you to consider the case that, you have a heterozygous parent, and you're looking at offspring that are crossed with, say, a homozygous other parent, another homozygous dominant parent, and then a heterozygous parent. So if we say, well, what's the likelihood of my offspring being heterozygous in the next generation? In this case, it's 50%. Why? Because I know I'm going to get a recessive trait from this organism, and I have a 50% chance of getting a dominant trait from this parent. So I have a 50-50 chance. And the same thing actually applies to if I have a heterozygous parent, but also a homozygous dominant parent. I have a 50-50 chance there too. So why? It's the opposite case. Half your offspring are going to be heterozygous, corresponding to the ones that get the recessive allele from the first parent, so 50%. And then we go back to our original Punnett square that we showed to say if I have 2 heterozygous organisms, well, that was in our IPRB. This is our first Punnett square that we looked at, where if we have 2 heterozygous organisms, 2 of the 4 cases are going to produce heterozygous offspring. And for that reason, in that particular case too, the outcome is 50%. Okay, so in other words, regardless of one's, trait for A, offspring with a heterozygous parent has a 50% chance of being heterozygous. Okay. So that's an important point that we want to make. So this is just me writing things down. But I have X, which is the number of organisms in the kth generation that are heterozygous for both A and for B. And what I want to do is to say, what is the probability then that X is greater than or equal to this parameter N? Now, let's imagine then I want to break this down to just one single case. So by the second law, where is that second law hiding? I'm going to know that the likelihood that, say, one offspring is heterozygous for both traits is equal to the likelihood that that offspring is heterozygous for one trait. Okay, so there is a hidden and here. And now I get to see, oh, well, where the second law is giving me independence is that the likelihood of an offspring being heterozygous for this and for this is the same as the probability of it being heterozygous for A times the probability of it being heterozygous for B. And here is where our insight is really useful, because now I know that for any particular organism, the likelihood that it's heterozygous for both is equal to the product of these two probabilities. And what did we say? Well, those probabilities in all possible cases are 50-50. So it doesn't matter what its other parent had. We know one of its parents will be heterozygous, but it doesn't actually matter what the other parent was. In all of those cases, this is equal to 0.5 for each of them. So it's 0.5 squared, which is just a quarter. So now I'm starting to understand how I maybe could compute this. I want you to imagine that what we're doing is we have 2 to the k trials. I'll write it as 2 star k since we're in Python. 2 to the k trials, and we want to know the likelihood that at least n of them are successes. And each one of those trials, the trials here we're thinking about the production of an offspring. And each of those trials has a certain probability, right? 0.25. So once I know that value, then I'm going to say, well, if only we had a way of computing this. And the fortunate thing here is that we do. This is called a binomial distribution. So if you want an example that goes back to things like grabbing balls out of bags, rolling dice, or say flipping coins. So this is analogous to flipping a coin with 1/4 probability of heads and asking the likelihood of at least n heads in 2 to the k trials. If you have seen something like this before, then you'll know, and this goes all the way back to, in a certain sense, Isaac Newton, because this is a binomial process, and we have a formula for this. And so I don't necessarily want to go through like exactly where the binomial process, how I obtain a final number here, because that's going to take us an extra hour to do effectively, but there are lots of good resources for this that I'll let you check out. But what it says is, in this case, my probability that x is greater than or equal to n, and I keep writing n as lowercase or capital, and I think you get the idea. In this case, it's equal to, I guess I've had lowercase n here yet. It's equal to the sum from I equal to n to I equal to 2 to the k of a bunch of possibilities for I. So what these values are summing all of the likelihoods that x is equal to I. So we're going to have a big sum of all of these likelihoods. Why is that okay? Here's where we get to apply this notion of disjoint events. It's not possible for me to have exactly 14 offspring that are heterozygous for both traits, as well as exactly 17. Just like it's not possible for me to flip a coin 100 times and get exactly 14 heads and exactly 17 heads. That's ridiculous, right? So as a result, these events are disjoint. And when I'm looking for this sum, I'm just going to write it as this. So then the binomial process is going to tell me what this probability is. And that's where we're a little bit outside of what we want to do today. So that sum then is equal to, I'm just going to make sure that this aligns here. I guess I needed a comment. That would be good. Okay. Is equal to this sum of a very specific thing, which is 1/4, and this sum is the same over all these values, 1/4 raised to the I times 1 minus that value that we have, which is 3/4, raised to the n minus I. In this case, it's 2 to the k minus I. Okay. And so we're going to have this value, we're going to multiply it by this value, and then we're also going to have another product here, which is the number of ways in which this can happen. So this represents, gosh, you got I heads in your coin flips, right? You had a coin that had a 1/4 chance of coming up heads, and you got I heads. Well, if you got I heads, that means you got 2 to the k minus I tails. The likelihoods of those were 3/4. But you also have a lot of different ways in which each of these outcomes could happen. It's not like I have to have exactly I heads come up first and then 2k minus I tails after that. So we need to multiply this by all the different ways that that could happen. And this is where we use a combination statistic that's going to tell us the number of ways. So that combination statistic is the number of ways to pick I heads from 2 to the k total coin flips. So there is a formula for this, which actually we cover in Programming for Lovers as an exercise, so you can solve this question there. It also is built into the math package. So at the end of the day, if you want to say what are the number of ways of choosing I objects from a collection of, in this case, 2 to the k objects, there's a formula there involving factorials, but I can pull this from a math package, so I don't need to worry about it. What's nice about it is you'll see that a lot of things came together. Independence was used here, and we also got to use the notion that if you want to say for a random variable, what's the likelihood that its value is at least equal to n? Well, in this case, we can say, well, that's just the sum of the probabilities that it's equal to I for all the values that are greater than or equal to n. Now we have it on good footing, and we understand what we're doing. And once again, as we've seen with a lot of these probability questions, when it comes time to actually solve the question, which is up here, taking all this and putting it into a function is not particularly hard. So the key probability that I care about, I'm just going to call that thing P, and it's going to be equal to 0.25. We're also going to need 1 minus P, and that is so common that we usually are just going to, so we have this term here is our 1 minus P, that's 3/4. That's usually written with binomial distribution as equal to 1 minus P. So it's equal to 0.75. Then we're going to say total is what we're eventually going to return. I'm going to start it at 0, so I'll return it at the end. And then I want to range over my population. So we're going to have our I. It starts at n, and it goes up to the population plus 1. So that's our 2 to the k. So these are all the possible cases that we have. Here's where I have my sum, in this case, of all the possibilities. So I'm saying I is going to range from n to 2 to the k. So what I represents is, for this particular outcome, the number of offspring that are indeed heterozygous for both traits. So the number of heads that came up in our coin flips. So I'm doing this sum, and then I'm going to need to take this probability. So I'm going to perform this ranging. And at the end, I'm going to say we're total plus equals something. So I'm adding the current probability. So I'll say, let's say current probability, or probability of exactly I. How about that? Equal to something. And after I compute this, I'm just adding that probability into the total. And in the meantime, I need to compute it. So here is where I get to use the formula that I had, which says that the probability that x is exactly equal to I is this thing. So it's p times raised to the power of I times 1 minus p, or q, raised to the power of the number of possibilities here, which is the population, minus I, times this combination statistic. So I just have to plug them in, and I've already set population equal to 2 to the k, so that makes things a lot easier here to write it, because I'll just say p raised to the I is our first term. We multiply that times q raised to the I, and then we multiply that by, and I don't know why I have an extra bracket up there, but we multiply that times math.comb, which is this number of ways from a population having that particular size, we can pick I elements to form a set. So that just comes from the math package, and we multiply it by p to the I, and we multiply it by q to the population minus I. I got the wrong formula there. Okay. Hopefully that's right. The good news is, again, the math was long and hard, but the code itself is quite short. So we're going to look at the terminal. And I could just click a button to run Python, but it's no fun doing that. It feels more fun to just say Python 3 lia.py, and we see 0.684. That's our expected number. Good. So I think we should be ready to comment our code. For parsing, go to our platform, download a data set. Take that data set, make it dataset.txt. And run again, and that should give us some output. So the output here is 0.326. And our data set here was, for this particular case, what's the likelihood after 6 generations? So you would have 64 offspring. What's the likelihood that at least 18 of them are going to be heterozygous? A problem that might initially have seemed like it wouldn't absolutely have such a short computation for it, and yet it does. Pretty cool. This is one I'm honestly, I mean, that's not easy. So I'm honored. I would never have imagined when we made this question. And maybe we should have tried to design the platform to reach as many people as we could. I don't think the intention was that this would ever be solved by 7,000 people. That's kind of impressive and humbling, because it's not an easy question. I do think there's some value. Hopefully all 7,000 of those people, when they solved it, thought, I see what this problem is trying to get at, right? Which is the idea of independence coming together with, in this case, a binomial process, and a little clever trick that the likelihood of any offspring is always going to be 50%. The likelihood that it's going to be heterozygous is always 50%. All that kind of coming together, and you need every single point of that in order to produce a nice, tight, efficient solution. So we're going to look at a couple more questions before we wrap up today. I think we've hit the peak of the mathematical side of things. We're going to have one more kind of mathy question, but when we're going to do it, it's not, what we're going to look at is not particularly, the trick of it is not like mathematically involved. We'll actually take a little bit of a step back mathematically with computing what we want to compute. Okay, and our problem is going to be prob.py. As always, let me know if you have any questions. Otherwise, we're going to keep going. All right. We're going to look at an introduction to random strings, which is the problem with ID PROB. And this is an easy question. I'll actually go ahead straight to the input and output. We're given an existing DNA string, and we're given an array that contains a collection of numbers. Those numbers are going to represent GC contents. So if there's an idea of the question, it's really to get at the idea that a lot of algorithms that we study in computational biology might say, I'm looking for a little bit of signal in this DNA data set, right? So if, for example, I was in upstream regions of genes, and I'm looking for a binding motif that's shared in those genes because they're all implied in the same function, or we've done ChIP-seq and we know that they're all regulated by the same transcription factor. So I know that there's something that this transcription factor is binding to, and I'm looking for whatever this word is. That's a very fundamental bioinformatics question. So if we're solving that question, we're looking for signal in these regions. And one of the things we need to be careful about, so something that, for example, if you had no biological background knowledge and you just came to computational biology and started working with DNA, something you could get fooled by is that some organisms, and we saw this in a previous problem where we computed GC content, but some organisms have a higher or lower probability, or should say frequency, of G's and C's compared to A's and T's in their genome. Sometimes it's more than 50%, sometimes it's lower than 50%. That means that the algorithms that we apply to these regions should be aware of this, because if you have an organism that is very GC rich, well, you're expecting to see strings with a lot of G's and C's in them. So you should make sure that if you see a binding motif in that region that happens to have a lot of A's and T's, you should make sure that that gets scored higher as a result, because something significant is going on there, right? So that is the idea of what we're trying to get at with the problem, is to prepare your mind to think about that sort of direction, to think about GC content being important. And in this particular case, we take a string and we want to say, if your GC content varied, what would be the likelihood that that string could arise due to purely random chance? That might be different, especially imagine this string has lots of G's and C's in it. If you have a low GC content, well, that probability is going to be a lot lower than if you have a high GC content. So this is an opportunity just to take that idea, play around with it a little bit, and put it into a function. So what we would produce at the end is the likelihood that if this were the GC content, you would produce this string via a random process, and the likelihood that this one would produce that string and this one would produce that string. So we're going to have an array of probabilities, but they certainly do not look very much like probabilities. And the reason why is because they're negative. And the reason why of that is that when values get close to zero when we're computing probabilities. You can imagine a long string. The likelihood of actually computing that string is probably going to be close to zero. And when values get close to zero, they often are going to get tough to differentiate and tough to store. This, I think, has gotten better over time, but it's a classic challenge in computer science, which is a lot of times we need very small numbers and we need to represent them very accurately, but we only have a finite amount of storage associated with each one. So in order to differentiate these and to avoid the circumstance where something is so close to zero that the programming language is just like, oh, the answer is zero, but it's not zero, it's 10 to the negative 30, right? So in order to handle that case, a lot of times we can instead take the logarithm of these values, because the logarithm has a couple of nice properties. For one, it takes values as the x value here heads towards zero. The values become more and more negative, so they head towards negative infinity. So I take values that are very small and I spread them out. But the logarithm also has a nice property, which is that if you take the logarithm of a product of things, we're going back to this overarching idea of being able to simplify a mathematical statement, which we saw with expected value, where we said, if I have two random variables and I'm looking at their sum and saying, what's the average value of this sum? Well, I can just say, what's the average value of one? What's the average value of the other one? Add them the two together, and that's going to produce the expected value that I care about. Here we get to use this concept of that you learned about if you've learned about logarithms, that if I take the logarithm of a product of things, the logarithm of xy is just the logarithm of x plus the logarithm of y. And you learn about that in a purely mathematical context, and then you never see an application of it. And so if nothing else, this problem is an opportunity to see the fact that you can split a logarithm as a sum over a logarithm of products. So taking the logarithm will allow these values to not just be very close to zero, but then that second fact is also going to be really useful. So let's come to our VS Code and allow myself to write what I just said, which is that for any base, we have this critical math fact you've probably never used, which is that the logarithm of x times y is equal to the logarithm of x plus the logarithm of y. Nice. So we're going to get to see it here. And if we think about how this is going to work for a single string, this is actually going to be pretty easy. So I'm going to have a higher level function that's going to take a DNA string, and it's going to take this list of GC content values, and it's going to produce a list of floats where it's the probability that if you have this current GC value, it will produce this string as a random string. But I'm going to also take the logarithm. So to implement this, I'm going to have my result list, and it's a list of floats, so I could be more precise about the type here. And that's what I'm going to return. And in the meantime, I'm going to say, let's range over each value in this GC values list. And then as I go, I'm going to say append the result of taking this for a single specific value. So here's where I'm going to call my subroutine log random string prob. And I'm going to give it the DNA string, and I'm going to give it the current GC content. Now, that allows me to think about just the single case of having a single string and a single GC content, and then returning the log base 10 probability that I care about. I'll use this critical math fact. I'll also point out that, the probability that at the end of this process, the string that we produce is equal to DNA. Well, that's equal to the likelihood that the first character is equal to the first character of DNA, and the likelihood, the part of this event is x1 is equal to DNA of 1, and we keep going until we finally say and the event that x of length of DNA minus 1 is equal to DNA of length of DNA minus 1. So we represented this probability that we care about, and I haven't taken any logs yet, but I've just represented it as the constitution of the intersection, I should say, of a bunch of smaller events. And now our process is generating a bunch of individual symbols that we're going to use to form this random string X. As a result, that's a lot like what we talked about with rolling 2 dice, right? Each of those trials is very clearly independent of each other, which means that when I have the probability of a bunch of ands, events that constituting the intersection with a bunch of ands, I can also say that this probability happens to be equal to the product of all these things. So I will say it is the probability that the first symbol of my random string is equal to the first symbol of the string I care about, times the probability that that's true for the second symbol, times, and so on, until I finally have the probability that X of last is equal to DNA of last. Even that's not going to get it close enough. So I hate using negative indexing. Ugh. If you like negative indexing in Python, what that means is we're looking at the symbol that's at the very end. Okay. So if you're a fan of this, at least it keeps me on one line. And now an important note is if I want the logarithm of probability of X equal DNA, that's important. So it's the logarithm of this product. And now I will get to apply the log of X times Y equals the log of X plus the log of Y. Now I get to use that fact because I'm going to have a logarithm of a product of a bunch of things. This term and so forth down to this term. After I take the product of all those things, if I want the logarithm of that thing, well, it'll just be the logarithm of this plus the logarithm of this plus, and so on down the line. That's nice because now I get to come and write my function that takes a DNA string as input and the GC content I care about, and it's going to return this probability of generating the string. After I take a logarithm, it doesn't matter what base I use. I'm going to use the logarithm base 10. Okay. So we're, in this case then, just going to write that function. And the only hard part of this, or the only tricky part, is to say, what is the likelihood? I just have to compute each of these probabilities. What's the likelihood that if I'm generating symbols randomly, what's the likelihood that the symbol that I generate is going to be equal to the current symbol of the string? And here's where GC content is important, because if the GC content is 50%, the likelihood that I'm going to get a hit for each of these is going to be 0.25. So that's why we added this little GC content wrinkle, and for the reasons that I talked about earlier, which is a lot of things are going to involve probabilistic algorithms, and so the identity of the thing that you're going to look for is going to relate to the GC content. So I should say, if I'm grabbing a G or a C, well, if the GC content is 60%, I'm going to assume that that means that the genome has 30% Gs and 30% Cs. I think that's a fair assumption. And complementarity is going to give that to me, because if I'm looking at the two strands together, I could have a lot of Gs on one strand. Well, that means I'm going to get a lot of Cs on the other strand. So I'm going to just say the likelihood that I generate a G or a C is the GC content divided by 2. The likelihood that I get an A or a T is equal to 1 minus that. So I can say it's 1 minus the GC content over 2, or 1 minus the probability of GC. Then I'm going to say I'm computing a probability. I'm computing it with logs. So I'm going to need a value to start off to be equal to 0.0. At the end of this, I'm going to return the total. And then I want to range over my string. So range over the string and determine the probability at each position of generating the symbol matching the current symbol of the string DNA. So I will say for each base in the DNA, I'm ranging over the string, which I called the string. And then I'm going to say, well, what is this current base? And I have a quick test in Python to say, is it a G or a C? To say, is that base in the string GC? Okay. And so this says G or a C. Else, I'm going to say this is an A or a T. If it's in GC, then what I want to do is, I could say, really, if I were computing the product, so the probability, that's what I would be interested in, right? Let's call it probability. Then I look at my product here to say, by independence, I'm just computing the probability of each of these trials. So my current trial has a certain probability, which says, well, if I've hit a G or a C, the probability is going to get multiplied by the likelihood of the current event. And that's prob G or a C. So if the GC content, again, is 60%, and the current symbol is a G or a C, well, I have a 30% chance, which is half of this. I have a 30% chance that the current symbol is going to match, and I take that 30%, and I keep adding it, or I keep multiplying it, I should say, into this probability that I'm building up as a product. So in this other case, I would say probability times equals prob of A or T. And now I've got this chain of products of probabilities that I care about. And at the end of that process, I would return the probability. And here is the point, that that's what we want, but these numbers are going to get really small for long strings because I'm going to keep multiplying like things that are like 0.2 or 0.3 together, and this number is going to get smaller and smaller and smaller. So imagine something that's like 0.25 raised to the 100. That number is going to get pretty small. 1 over 4 to the 100, very, very close to 0, going to be very challenging to represent a number like that. So we're getting close actually to the likelihood of picking one atom on one hair of your body out of the entire universe. So that's not a big probability. And so as a result, this is where the logarithms are important. So what we want is the log base 10. And Python gives us this, math.log base 10 of the probability. But the point is, and this is a critical idea of why the logarithm is useful here, is that at this point, it's too late. If I have something that is too small, it's essentially indistinguishable from 0, and I've run out of the ability to distinguish it from 0 internally in the computer. Well, at this point in time, I'm cooked. Taking the probability of that is just going to blow this to negative infinity because the computer has already said it's so close to 0, I'm storing it as 0. So that's not good. And here is where we are fortunate because I'm not going to compute the probability first and then take the logarithm. I'm going to start with a total. And the total is what I'm going to return. And the total is going to use my math fact that the logarithm of this product, or probability, is just the sum of logarithms of each individual part. And what is each individual part? It's each of these values. So the current likelihood that I matched is the probability of GC, or the probability of AT. Instead, I'm going to be adding math.log10 of that probability. And I'll do the same thing in the case that I have an A or a T, which means I take the logarithms as I go to make everything work out, and I don't have to take the logarithm at the end, and I simply can return the total that we've obtained. Now I can run this and say Python 3, prob.py. And we see we have a bunch of values. Now, those match, I hope, the values that we see on the platform. I don't know. I hope so. Seems like maybe they don't. Yeah, they don't seem to match. So that's not good. There's another issue that I noticed in this particular case, which is that we need to round our output. And I will probably, when I will post the code with this cleaned up, but there's two issues, and I know what one of them is. So number one, let's look at the easiest thing we possibly could fix. So when I produce my values, I like having an exact value here, and I probably should fix it in like at the print stage. Generally, there's a principle that you don't want to round something and truncate your data before it's like until the time that you care about doing that. And in this case, I like storing the values as long as they need to be. This is fine. I want my data to return these complicated values, and then I can produce that output that's truncated to the user. I'm gonna have to change a lot of stuff to do that. So I'm going to instead, when I append the value, when I call this function, I'm going to round this to 3 decimal places, and I can just do that with round. Okay, so when I run this, now that'll get me down to something that's going to match the parser on the website. Better way of doing that is handle it in main, but for now this is okay. The second problem is that my values don't match. And if we have, there's not a whole lot of, we have modular code, there's not a whole lot of places that this could go wrong. This just seems like it's okay. This idea is right. At every point in time, I'm going to take the total, and I'm going to add in the logarithm base 10 of the current probability. I guess I also could have the wrong base, but I don't need to go to the website to tell you that it is indeed math.log10. I should be taking the logarithm base 10 in each case. The total should be 0. So I could debug this or write a bunch of test cases. And if I wrote a test case, I would probably see that my likelihoods even for short strings are going to be wrong. And that's because I have the probability of C is equal to the GC content over 2. So if I have GC content equal to 60%, this should be 30%. That's good. But the probability of an AT should be 20%. Why? Because, well, the GC content is 60%, the AT content is 40%, and then an A or a T should come with probability 20%. But that's not what I have here. I would have set this equal to 1 minus probability of GC, so the probability of an A or a T is now equal to 70%. So that's not right at all, okay? And instead, what I want is to take 1 minus the GC content. And so in the case before, the GC content is 60%. This would give me 40%. Okay, 1 minus 0.6 is 0.4. And then I can divide that thing by 2 to split it over the cases of an A or a T. And now when I run it again, I get values that sure look a whole lot like the values that match the platform. So I think we're in okay shape. And I haven't solved consensus and profile. So just magically pretend that I've got this correct. And we are going to solve our final question. And after I solve the final question, I'm going to come back and solve this question. So trust me that we've done it. If you don't believe me, well, you'll just have to stick around. We're like a consensus and profile. So I'm going to delete this for now. And this is CONS if you're following along. Let me know if you have any questions. Anna, you said you thought prob AT is not correct. And I figured it out myself. If you beat me to that, good job. I missed that somehow. I think it's probably, unless I'm wrong though, unless I just wasn't paying attention, it's always what normally happens, which is right as I fix it. Somebody's like, oh, it might be this. I was thinking that for the last 18 minutes. Sarah, welcome. Good to see you. You joined late. I'll forgive you too. It's all right. So we're going to look at cons. And this is a classic like bioinformatics algorithms type question. It's actually in our textbook. So, or functions that you need this stuff are in our textbook very early on. And this is inherently related to motif finding. Or I've got a bunch of regions. What's a word that they all share? Which I guess is why this problem is a prerequisite for what we already solved, because I told you why you might be interested in this question, and here's an example of that. So we've got a bunch of sequences, and these sequences could perhaps be like regions of interest that might be similar to each other. So we're going to work with that. And this function, this problem, if we were doing it over again, maybe I would split this into two problems, right? It's good to have things that are modular, but this problem is going to ask for two things. So we'll get started here shortly. Our final question is going to be CONS. All right. CONS is our problem here, looking at consensus and profile. A classic bioinformatics question of having a collection of strings, presumably DNA strings, but perhaps from another alphabet. And we would like to compare these to say we've identified perhaps strings that are similar to each other. And a lot of times in bioinformatics data analysis, what you see is some columns, when you line these strings up, are going to be more conserved. Some are going to be more variable. To say from an evolutionary perspective, some of these positions really are not allowed to change very much, but others are. So our third column here, for example, has a lot of G's and one C. And our second to last column has a bunch of C's and one T. But other columns are allowed to change a bit more. So this column, for example, has 4 C's and 3 G's. So the first thing that we might do is make a profile matrix that simply ranges through all the strings and counts up the number of occurrences of each symbol in each column. Fair enough. And then another important consideration is to say, well, what's the most frequent symbol in each column? So once we have that information, we can produce a consensus string that tells us what the most frequent symbol is in each column of this alignment, so to speak, of our strings. So what has the maximum value? A, T, G, and so forth. And that's how our consensus gets formed. So it's a straightforward question. There's some parsing, which I'm going to assume that we already have, which is going to process a FASTA data set, giving us a collection of at most 10 DNA strings that we see. And then the problem is going to ask us for both of these things. Now, constructing the profile matrix, once I have that, it will be easy to form the consensus. The output is asking here for the consensus first, and then for the profile matrix. So we're going to print them in reverse order, but the idea is we want both of these things. So we simply need to write both of these functions, and this is an opportunity for us to see, a little bit more about essential algorithms for string analysis. Let's look at profile matrix first, since we can do them in either order, but until I know how the profile matrix is formed, it doesn't probably make a whole lot of sense to write the consensus string function. And in this case, I'm going to say, well, let's assume that the data are good and the sequences have equal length. This is going to be very annoying and impossible to solve if that's not the case. So I could imagine that we could write a function to assert that these functions have equal length and maybe produce an error if they don't. But what's nice about Rosalind, at least one aspect of what's nice about it, is that even though there's a lot of tricky things with input and output and difficult to impossible mathematical challenges that I provided very little intuition or additional information about to try and get you to uncover as Easter eggs. Even though we have all of that, we also have data that always come in a nice format. So you don't have to write all these assertion functions to make sure that everything is okay. And I'm just going to say whatever the length of the first sequence is going to be the length of all of them. And then I'll establish a profile matrix that's going to be, well, it's going to map strings. So every row of my profile matrix is going to correspond to a letter. So A, C, G, or T. And then the row is going to be a list of integers. So it's always good to think about the types. So the row here is just the number of counts that I see at each column. And then I'll say this is equal to some dictionary. And I can use a dictionary literal. It's nice. So I'm going to say, I'm going to start off A as being a bunch of zeros. In particular, how many? Well, the length of each string. So the number of columns in my profile matrix should be equal to the length of all the strings. So I'm going to have n zeros corresponding to A. I'm going to have n zeros corresponding to C, and so on for G, and for T as well. And I'll add a trailing comma. A lot of languages demand that. Python doesn't. That's all right. Once I've established my profile dictionary and I've made this thing, now I'm going to range over all the sequences and just add one every time I see a symbol to the appropriate spot. So I need to range over all the sequences. The first sequence is going to be called seq, and it's just a string, but we call them sequences in bioinformatics. And then once I'm in the current string, I'm going to range over the length of that string, okay, which is really just n, okay? And then I'm going to say, okay, what's the current symbol? So what I care about is the current symbol, which is seq of I. So if I want to be very precise, I could write that out and say that the current symbol is equal to that in that string, whatever is at the current position. I is also going to be useful. So you're right, like, why didn't I just range for symbol in seq? Well, it's because I is useful too. And if we look at the profile matrix that we have before, and we think about ranging, so I'm ranging over this second string, and I'm at position 3, so I'm looking at this C. That means I need to go into the profile matrix, and I need to access C, but I need to add 1 to the value at index equal to 3. So this value is going to go up by 1 at that point in time because it corresponds to the current index that I see. So that's why the index I matters here, because what I want to set is look at the profile matrix, go in and say, well, the profile matrix is mapping strings to lists of integers. So it's profile of the current symbol, this temporary variable that I don't need, but that might be useful to think about. That current symbol, and then I'm going to need to go into its, so if it's a C, I need to go in and set its appropriate value to have 1 extra to be incremented by 1. So the value I care about is at position I. That's what we said was why I is important from before. So go in and access for that particular list, look at the element with index I, and simply add 1 to it. So here's where our increment happens. And that's it. That's all I do. At the end of that process, I return profile. So now I have these 2 tasks. I've written one of them. And then it's just a matter of producing the consensus string. The consensus string is going to take the profile, which is a dictionary mapping strings to lists of integers, as we've seen, and produce this consensus string that takes the most frequent symbol in each column of the profile matrix. A completely separate task. So here is where we'll say the result that I care about is going to be a list. And it's going to start off as a list of strings that's equal to an empty list. The reason why is because I'm going to generate my consensus string as I go. And we've seen that the best practice for this is not to make a string and then concatenate. It's to make a list of symbols, and then at the end of this process, join them together. So minor point that we've seen in a couple of different problems already as we've gone through this. And then it's just a matter of saying, well, we want to access, to know how many elements are there in each row. So I'm given the profile matrix, which is just a collection of integers corresponding to A, a collection of integers corresponding to C, and so forth. And n can be obtained as the length of these. So I can do something that for a very generic case would absolutely not be kosher, and this function is not going to extend very well. But I think I can fairly say whatever it is when I access the profile of A, take the length of that thing. This will tell me the number of columns in the matrix. So that's useful. And once I've done that, then I can say, then it's just a matter of saying, range over all the columns and take the most frequent letter. Okay. So for I in range over n, I'm going to say there's a critical point, which is all ties broken by the first symbol lexicographically. So if you had, if you have 5 C's and 5 T, then you pick C. Okay. So as long as we do the ranging appropriately, that's going to be okay. A better autograder, a more modern autograder that's not built off of this platform of randomization, would just run this on a bunch of test cases, and each time that it grades it would just say, well, does your consensus, is it might be different to what ours is, but does it at least have the right properties? Meaning, if there is a tie, you can pick either a C or a T here. But we're going to make this extra demand in order so that it matches it exactly, that you always pick, if you have a tie in the profile, as who is the biggest element, there's always going to be the first symbol is going to win. So I'm going to start by saying my best base is going to be equal to A. And then I'm going to range over all of the bases. And I'm going to see. So for base in ACGT, and I actually don't need to range over all of them. I guess I could just range over C, G, and T, but that's going to look kind of weird. So that's an optimization I could do later. But I'm going to range over all the other letters. And I'm going to say, does the current, the current nucleotide, by base I mean nucleotide, is the current nucleotide, or does it have a higher count than the best one? So let's make that valid English. And so how I can answer that question is to say, is it the case that when I look at the profile matrix, and then I say, look at the profile matrix of the current base, and I need to know that I'm in column I, so this profile matrix would give me a list of values, but I'm only interested in the value that's at the correct column, which is at column I. Is that better than what happens when I look at the profile matrix and I look at the best base and I look at its value in column I? And if that's the case, I want to update the best base to be equal to the current base. So I'm going to update it. So this shows you why I don't need to range over A, C, G, and T, but that's okay. And if that's the case, then I update that. I don't need to update anything else, because at the end of this process, after I've ranged over all 4 nucleotides, I'm just going to take the result and I'm going to append to it the best base that I found. So that will produce the list of strings that I care about. And once I've got that list of strings, I'm just going to join them together into a single string, because this is the optimized way to do it, so we don't have a bunch of repeated string concatenations. Now I can go to main, And I can run my code. And it gives me an output, and that, well, I probably should see if that output's any good. That looks pretty similar to what we had before. It seems like I'm counting, like, what's the number one thing we want to worry about? You know, we have 7 input strings in our sample. The values in all the columns should be equal to 7 when you sum them up. Shouldn't have any negative values. And the consensus string should make sense. So an A is the consensus value in the first column. A T is the consensus in the second. G is the third. And so everything seems to be in order. So I should feel comfortable to say, you know, we have our parsing that is happening automatically for us. And let's then download a data set and put it into our File Explorer. And I want to name it data set. And now when I run this, it should go to output. I think it went to output. And so in this particular case, we have a nice long consensus string. You see why we wouldn't want to do a bunch of concatenations. And there are our values that we see. Okay, so pretty good. I'll copy all this over, and I could also upload it as a file, but that's not too much to go on the clipboard. And I submit. And we see that we got it right. And we got string algorithms badge level 2. Most importantly, it's suggested that I go back to this question that we already solved. So now that we have solved the consensus and profile problem, I'm now going to be able to do what I would like to do, which is also solve my random string problem. So I've downloaded a data set for random string. And this is PROB. Everything should be in order in terms of, I need to comment out that. I got my data set for PROB, and I need to name it dataset.txt and run. So there's my sample. Here are these logarithms now that are much more negative, but my data set, you know, the string is quite a bit longer now. And if I were actually computing probabilities, these numbers would get really, really close to 0, but because I'm computing logarithms, everything's okay. And so let's see what our output is here. These are all those logarithms, which are just values that are not even as big as negative 100. But think about what that means. So this is saying it's about like 10 to the negative 83 would have been our probability. Pretty tiny. But now it's just a value between negative 100 and 0. And I can submit it. And we solved the problem. Yay. Okay. So notice what we're getting suggested. Speeding up motif finding, matching random motifs. I had talked about this problem in the context of finding motifs. And, you know, what are these strings that are similar to each other that are lurking in biological data sets or in DNA data sets as a hallmark of, you know, a very classic problem that we look at in bioinformatics. So that's a great plug. I mean, that is chapter 2 of our textbook in bioinformatics algorithms. And then the whole chapter is available for free online. So I probably should just put a link to our website in the chat. But I will also, we will also have this as a topic in Great Ideas in Computational Biology, which is coming soon. So that's a great plug for both of those things, because we will cover, you know, this concept of motif finding in both places. In the meantime, I'm going to be solving some of these problems, so you'll get a double dose if you like. You'll get a chance to see them in the context of us solving related questions on Rosalind too. So there's something for everybody. That's also a great reason for you to sign up for our mailing list and get on our Discord so that you stay in touch about not just these Rosalind streams, but the other stuff that we're going to be doing. So make sure you do that. Check all that out in the description. And join Philomath if you want some even deeper connection, as well as to do things like you get a free ebook, and there's the opportunity to complete projects on programming for lovers. And we're going to let you know about other stuff coming too. So we've got plenty to do. If we look at our tree list view, we see we're starting to get there. You know, we're starting to enter the problems that maybe only a couple thousand people have solved. So we're becoming survivors. And then we're going to hit a point, I think, as you see, where, I'm impressed this many people have done this many problems. But we're going to hit a point where the problems are going to become challenging. They're going to require us to think about algorithmic paradigms like dynamic programming, how we work with evolutionary trees. We're going to have to do more complex things with counting, like how we're going to count things. And here is where a lot of these problems became kind of mysterious, right? They're intentionally opaque in certain ways, because if you do get them right, you really feel like you've figured something out that's amazing. So we'll have an opportunity to see that. As you see, I think in terms of what a lot of people have done, we're starting to get beyond the basics. If we look at our tree, we're working our way down in the tree. We've seen these was the absolute fundamentals. We've now almost cleared out everything at this level. So we haven't talked about this problem just because we haven't needed to look at networks. Notice I mentioned something about motif finding. So what we just did is going to set us up a little bit for motif finding. It's a nice natural transition. And then we're going to keep moving through the tree and talk about things like, oh, this is Catalan numbers and RNA secondary structures, and we need stuff like dynamic programming to even address stuff like that. So we've seen a little bit of that actually with our Fibonacci sequences, but we're going to look at constructing networks, constructing trees, counting matches, doing all kinds of fun stuff. And all that is coming soon. So we'll do it next time you see me. We're going to keep working our way through the tree. Make sure to like and subscribe. You can sign up for notifications. We've got a big schedule of all our videos that are coming, so you can see exactly which problems we're solving on what day. Like, click to get notified about that, just so that you stay in the loop. Hop on our Discord, hop on our mailing list. I'll see you next time. Can't wait. And until then, happy coding.